Hi, this is for Ian. So the motif, the patient you showed, that's the lurcher motif.
Yeah, it's the exact same site we've got the equivalent
In front.
Okay, yeah, it's the exact same position, the exact same.
Right, which was right, as you know, was like originally identified in Delta 2 in animals, but that has variants that pop up in GRIN genes and GRIA genes. And I don't know off the top of my head how many different subunits of both GRIA and GRIN genes have rare variants in the lurcher motif.
All four GRIA variants do. There has been, I think there's a mum here with a GRIA1 at that site. We looked at the GRIA2, but there's also a GRIA 3 and 4 as well. I can't speak for the NMDA receptors, but I'd be surprised if there wasn't a variant there. But yeah.
Okay. Sorry, I have a question. I'm actually going to be obstructive to the program, but I have to fly back, and I just wanted to thank everyone. I just want to say something. It's a bit naughty of me, but what I would have said in the general discussion is that we are very open to apply our EEG technique, the quantification technique, for any trial, any EEG out there. We can apply it even to single electrodes. So if anyone wants to use it, or postdoc or prospectively, we can reach out, and we really would like to start a sort of collaboration to facilitate that quantification. I want to say, and thank you very much. I'll use L-serine for breakfast from now on every day. Yes.
Thank you very much.
Well, I have two questions. One for Silvia. I really would like for you to be a PhD in my lab. That was a fantastic presentation, beautiful.
Thank you.
And really, really well done in there. Question, because it has come up yesterday, Amy, Vladislav have shown sex differences. Have you looked into that at all?
In larvae, they don't have sex differentiate yet, so we don't have that problem at that time point that we are looking at them. Now we are planning in the lab, because it's not set up, and it's not only my fault, but we are setting up to do it with juveniles and with more adult fish, mainly to test autism-like disorders and that kind of stuff that we cannot do with larvae. And in that stages, we are going to take into account the sex, but now we cannot do it because they don't have the sex defined. So it's not a problem in these stages.
Okay, maybe it's anyway a wider discussion in the Q&A or in the roundtable afterwards. Shani, one quick question. Why did D-serine not work whilst L-serine did work? So, generally speaking, because you have a big of heterogeneity across patients, no drugs.
So first with the D-serine, we had a higher toxicity, so we had to reduce the concentration. So the L-serine is around 0.5 millimolar, and the D-serine is around maybe 2 micromolars. I'm not sure why. We haven't tried to analyze. It was something that we saw, like with D-serine, it was much more toxic. So maybe the fact that the concentration was so much lower, maybe. And also it didn't completely not work, but it was not as significant as the L-serine.
Yeah, but evidently the L did not convert into a D, because the system does not allow for that, I would assume, no? The conversion of L-serine into D-serine.
It happens in the neurons. I don't know if we haven't in the astrocytes. We have astrocytes in the dish. We have. So when we differentiate, approximately 15 to 20% of the cells in the dish are astrocytes, and they we have progenitors, and most of them differentiate into neurons, and some differentiate into astrocytes. And we haven't checked, for example, to see if we have levels of D-serine in after we put L-serine.
Okay, good. Thank you.
May I will ask you, the differentiation, the changes in development and the effect of drugs was quite amazing. I wonder whether it was specific to cultures that you use in transformation from non-mesenchymal cells or fibroblasts, and whether you see something similar in neurons when you have, let's say, hippocampal neurons, and if you put these drugs to normal neurons, whether you see similar delayed or change in the development of synaptic transmission and excitability of these neurons?
So we haven't done this experiment. Or maybe we have, but not as robustly. We apply them only to the mutant, mostly to the mutant neurons. We don't know. It would be interesting to try after all this L-serine. Everyone's talking about maybe try on the control neurons to see if maybe the networks become even more connected. But we haven't done it.
Dr. Stern, you already read this question. I'll just read it out for everyone. Could you say a bit more about cell type composition of the cortical cultures? Do you include both glutamatergic neurons, GABAergic neurons, and astrocytes?
Yes. So when we do cortical differentiation, we get approximately 10 to 15 from the neurons. We get approximately 10 to 15% of the neurons that are GABAergic positive. We do staining for GABA and they're positive. And approximately maybe 75 or 80% are glutamatergic, and the rest are we haven't characterized. And approximately 15 to 20% of the cells in the dish are astrocytes. We can be very specific, like with the differentiation. We can differentiate into other types of parts in the brain, like we did with the Parkinson's. We did dopaminergic neurons. And we also plan, also this is the first stage, the cortical, but we may look at, for example, at the different glial cells for this. But right now we have the cortical cells.
And another online question. This goes both for you, Dr. Stern, and for you, Silvia. Do you plan to apply your approach systematically to a larger number of variants described in the literature or in trials? This would enrich our knowledge on genotype, phenotype, and functional relationship, and it can help patients, parents to understand their diseases better.
Well, in our case, we really are looking for, not now because it's quite complicated, but the best that we could have is to have humanized models of zebrafish in which each model has one mutation of one patient. And the NMDA receptors are really well conserved, and probably for most of the mutations, we can have the similar, not the same variant in the zebrafish, but it's not going to be exactly the same. But yeah, it will be amazing. The thing is that we are still characterizing only the knockouts. So it will take a bit of time, but ideally, and if we can do it, it would be perfect to do that and to have one fish for each patient.
And just to follow up on that, so what would it take for you to actually get there?
First, to finish characterizing the knockouts and to really know if I think it's worth to explore it, but to be sure that we are looking for something that it makes sense. And once we have it, to have a stable line of fish that are laying eggs and everything, it takes for 6 months once you start. It's not that much, but I think we still need to work first in the loss of function general models before going to the patients. But I hope at some point it arrives. It will be really nice.
So for us, we may increase, not by much. I think the iPSCs are relatively expensive to grow, but it's easy. So it's like the compounds are relatively expensive, but we take blood cells from the patient. We have a protocol, and eventually we have neurons with the exact same mutation. So we may increase by a bit the cohort size.
A quick question that kind of tries to fuse a little bit what we're doing here with the GRIA, because we were discussing that some of the symptomatology that we see in GRIN, we also see in GRIA, and how much of that could be the influence of GRIN and the AMPA and the NMDA receptors kind of not working well together. And we're wondering if, this is something that, I don't know if it will be easier or no for those of you who work on either animals. I don't know if there's a GRIA zebrafish type of gene, or for example, in your case, Shani, that you have already the brain organoids. If these interactions are related to the effects, I know you cannot measure behavioral things, but just the interaction between them two in specific mutations might lead to make you think, oh, could this, what is happening, with the AMPA receptors in this GRIN disorders that we already studying. So I don't know if we can kind of combine the efforts to try to look also at from the GRIA side also.
I mean, I would say that the majority of symptoms that patients see are directly due to the act on the receptor. This interaction is important, but I think if this was the only problem, then I don't think our patients would be anywhere near as sick. So it's and this is something, I mean, this is kind of why we want to simulate it before we do anything else, because it's possible that, I mean, it makes sense in the cartoon form. It's possible that actually the magnitude of these effects is actually relatively small in terms of what changes with a variant. And so, yeah, I would say that at least we want to get our modeling sorted before we go too far. But the effect won't be 0.000. Now, if it's 0.001, okay, it's an effect, but maybe it's not so important. But that's kind of what we want to establish with our modeling.
Yeah. So for us, I mentioned the IQ-SEC2 model that we have, which the IQ-SEC2 gene relates to the AMPA receptors, and eventually the neurons have changes in expression, both of the GLUA2 and also surface expression changes. So, and when I look at the IQ-SEC2 and our GRIN2B, there are many phenotypes that are shared. We have this early hyperexcitability, and then later they're hypoexcitable with a reduction in their synaptic activity. And also, if we look at the patient's symptoms, they have autism, they have epilepsy, and intellectual disability. So I think that it would be interesting, like to try, like as you mentioned, the PAM or the modulators for the AMPA receptors on the GRIN2B and vice versa.
Yeah, so, yeah. If you have the system, then it's kind of, it's only a model. It's only ever a model when you simulate. So if you actually have an actual neuron, then that's clearly better. Yeah.
And while in our case, one of the things that we are planning to do the following months that I mentioned, but I didn't say the name, these integrative studies that we're planning to do is what we name transcriptomics. That is that what with the mutant fish and with the control fish, we analyze the expression of the different genes related with an NMDA receptor somehow. The idea of this is to find some hits, it's what we name hits, some proteins, some genes that are somehow upregulated, downregulated, upstream, downstream of the NMDA receptors that are influencing the NMDA activity. And I'm sure that when we do that, some GRIA genes are going to appear for sure. Or maybe some GABA genes or something that modulating the synapse and how it's working would help in some of the symptoms. I don't think that we will find something that will help with everything, but yeah, for sure, I think that they are going to appear, these GRIA genes in these transcriptomic studies for sure. So let's see what happens.
Yes, I have a question for Ian. So it was very, very nice presentation, and all of them. And I was wondering about the functional studies. So you have only one combination of auxiliary proteins together. So my question is to know if you consider another auxiliary proteins, because depending on which GRIA gene is affected, and the clinical symptoms, as far as I know, for the AMPA receptors, they have different combinations. So perhaps you could drive and try to adapt a little bit, or is it too complicated?
I mean, I'm expecting us to maybe go to town a little bit more on a few of the variants. Certainly, if you were to ask most people in the GRIA audience, some of which are also here, what is the most prevalent receptor in the hippocampus, for example, and obviously the hippocampus in seizures is a GLUA1/2 heteromer along with gamma-8 and cornichon-2. So it's already a bit of a lump of stuff. So if you have a variant in GRIA1 or seizures are a particular symptom you're interested in, then making sure that the variant has an effect in that combination certainly makes sense. Actually, the Godzilla channel I told you was something kind of interesting. We always expect the TARPs to make the currents bigger, but we've now had 3 variants where we expressed it as a homomer with a TARP and we saw nothing at all. And in all 3 of the cases, when we then expressed it without a TARP, we saw receptors with really large gain of function. So in those situations, we would have had it completely wrong if we didn't take the TARP away. And so, yeah, certainly for the first screen, it's kind of really important just to get as many as possible and choosing one condition. But there are certainly situations where you want to refine for sure. But there's only so many hours in the day. So yeah.
And second, I totally agree. So I won't say that it's a how did you call it? The semi-precision therapy. So I really love this concept.
Oh, okay.
But I
you
think
owe me
it's not
10 pounds.
as
You owe me 10
semi.
pounds.
So it's even more precise because you know much better the signaling and the relationship between AMPAs and NMDA receptors. So somehow it's a more precision medicine. I will change for MPM. Perhaps.
Right. Yes. Yeah.
But I totally agree that this is a way to proceed instead of repurposing to find signaling cascades and modulators.
Yeah, definitely.
So what will be, let's imagine your favorite model. Would you use her iPS cells or zebrafish models or
I see. To ultimately do the experiment if it looks good. Well, I mean, the main thing I'm excited. I mean, I want to try it just because we've got nothing for Greer loss of function at the moment. There are a whole load of PAMs that have been developed, but none of them ever got approved. There's nothing you can actually give to a patient. And, you know, at least half of the patients with Greer are loss of function, and there's kind of nothing to do. So if at least the L-serine is something to try and boost the system. And so that is what I would be most interested in trying. But then getting the model for that is kind of slightly, you know, the L-serine needs to be metabolized, and so how you do that within your system, I guess you've kind of addressed this. But that is kind of that in general, that's the principle. In terms of what the experiment is, then you know, we haven't really thought too hard, but we have started to do a bit of work with our PSCs, so I'm hoping that could be something we could do.
Okay, so thank you very much. In any case, I think that joining GRIA researchers, clinicians, is crucial. Thank you.
It's also good to write a section on our webpage about it. So that's
yes, yeah, and it will be really thought through, unlike this. So,
okay, just a comment about the L-serine. According to Shani Stern work, we saw drugs that might be more effective than L-serine. So I want to mention that. And I have another question about the zebrafish model. Other examples that it's a very nice model. If there are indeed chemicals that were found and developed into the clinic based on this model.
Can you repeat, please?
Other examples how the zebrafish model contributed to the development of drugs that became a drug.
The examples that I have more next to me is that the ones that I know from the clinics a few years ago started, I don't know if it's a startup, but what's the name of this kind of stuff, but another company related with the clinics that it's related with cardio regeneration. And they tested already several cardioprotective drugs or therapies that have already gone to mice, and they are working with patents, and it's going to patients directly. And there are several studies that I don't know all of them for sure, but there are studies where there's clearly a relation between the dose and the toxicity in the zebrafish and how this can be moved translationally to patients. And it's this conservation of the metabolism of the drugs, of the therapies, the metabolism of the animals is really well conserved. It seems weird, and I know it's kind of weird, and it doesn't work for every therapy. But as far as I know, in cardio, it works pretty well. And in neuroscience or neuro stuff, it's being quite complicated not because of the zebrafish, but because in most of the clinical studies, the therapies need to drop when going from mice to humans. So we don't know exactly what's happening there and how can this be translated from zebrafish to humans.
Again, guys, I have a question about so yeah, like what you were saying, when we look at mice, there's a high clinical failure rate when you try to then translate the results to humans. So is the advantage to the zebrafish just like the cost benefit and the expediency of their development? and then kind of in line have either in these zebrafish or maybe also in the neurons and brain organoids. Is there a potential to mix some candidate drugs? So like, if we mix a PAM with maybe something like a pharmaceutical drug or something like that to see if they can accentuate each other?
In the case of the zebrafish, the thing or the positive thing that I think that we have when working with them is that you can test in a fast way more therapies or more candidates in a fast way, not to spend that time, that money, and those resources in mice, because if it works in zebrafish, very probably it will work in mice. So you can have this translation in an easy way, and you are not spending that much time and money just testing it only in mice.
So you would describe them as a bridge between the, like neurons and mammals, because for the cost effect.
Well, the thing is that the good thing is that when you are working, and I've been working with mice and with cell cultures, primary cell cultures, and with organoids and with neurospheres, and I'm trying to touch everything I can because I like to do stuff. The advantage of working with animals, in our case with zebrafish, is that you have the whole system. You don't have isolated brains, or you don't have the isolated CNS. So you can take into account somehow, because it's not a mammal, but it's quite similar, not only what is happening or the effects in the CNS, but how the rest of the system is getting involved in this reaction. And about the double treatments and combined treatments, I think it's a possibility, but again, you need to take it into account into a system perspective, because if you're only seeing, you always start seeing only the cells or the type of the cells that you're interested in, but you cannot stay there because you're not taking into account everything else that's happening in the body. But,
yeah. So for us, it's relatively easy to.
Yeah, it's on. It's on.
So we grow the neurons and the organoids in 48 or 96 well dishes, and we have like a bunch of conditions. So it's relatively easy to try combinations of drugs. So. Yeah.
Thank you. But you guys see potential in that possibility?
Yeah. Yes, we do. And I also mentioned, I think also that we another lab in Technion mentioned that L-serine may need to be supplemented with other amino acid that we're now checking it. So it's very easy to take the L-serine, put in another amino acid, and so forth. So
okay, thank you guys.
I have a question. Are you planning on doing so? Or I know it's a possibility, but are you planning on combining drugs and testing in your models?
Yeah, we are planning to. Yeah.
So what's the time kind of?
So I would say months. Like we have right now we have 3 that we already have neurons and organoids, and we have tested a bunch of drugs, and we have 2 that we just have now, young neurons, young organoids that we just started with the testing. And so, it's within months that we will try combinations. Yeah.
Thank you.
We don't have any more questions for our panelists, so thank you very much. And if you don't mind, just stay where you are. We're just going to do a quick roundtable discussion, including all the participants. But thank you very much for your presentation.